so much. So I'm going to be talking a little more about Fernando's work focused on DNA barcoding. So in case you're not familiar with DNA barcoding, is focusing on a fragment of DNA that is variable enough in order for us to be able to identify to a species. However, identification of species are much more complex than a few hundred base pairs of DNA. So just to give you an example of how a project that started as DNA barcoding, so this particular project that was published in earlier this year started when Tom Iliffe came to me and asked, can you sequence these shrimp from Mexico? And once I started to look at the data, I already started to notice some of the red flags. What eventually became a multi-institutional collaboration, eventually led by Lauren Ballou, who's now at Florida International University. This study highlighted some of the issues that we encounter with working with species that are morphologically similar. And so over the years, what occurred was misidentification of species, which in turn led to differences in terms of the phylogenetic relationships that were being published over the years due to these misidentifications. So the study also highlighted the importance of the inclusion of specimens from type localities, that is, the location of which a species was first described from. And so even though some locations that probably haven't been visited since the original description and sometimes are impossible to pronounce, as you can see, this eventually becomes an important means to clarify species identity. So the inclusion of specimens, so that was for Tifladia campeche, here from the green star in the middle there. What this study showed, not only looking at what started as that barcoding, but also looking at other gene data, we were able to show that Tifladia campeche appears to be basically genetically similar to Tifladia pearsi, another known species. So the analysis also showed that there were 5 different lineages. So those 5 lineages not only revealed that there are 2 species that were unknown to science, but it also began to give us a picture with regards to the distribution of species due to the broad collection that was conducted over the years. And in particular, looking at Tifladia zillamensis, you can see that the distribution of zillamensis is all along the coast, so specifically ancholine, okay? Whereas the other species can be found throughout the freshwater lens throughout the aquifer of the Yucatan. So in addition to that, we also included environmental data. The inclusion of environmental data also allowed us to be able to identify zillamensis specifically as euryhaline, which at the time, the species was only thought to occur in a single cave, but also from the saltwater layer. But as you can see here, specimens were collected from nearly freshwater all the way to over 30 PSU. Now, the reason why I'm talking about this particular study is what really needs to happen when we're trying to identify species. It's not just about a barcode, but this is all the work that is needed and where we need to integrate multiple tools and use these integrative approaches, but also the importance of these type localities, especially when we're dealing with species that do have a conservation status. So Tifladia pearsi and Mitchellite, they're considered to be vulnerable and federally listed in Mexico. And as you recall, Campeche had only been reported from its type locality. However, the genetic data showed that it's possible that it probably doesn't exist, and it is in fact Tifladia pearsi. What the study also highlights is that now we have 3 species that have unknown conservation. And as you saw, Tifladia zillamensis is only occurring along the coastlines. And what's along the coastlines? Lots of anthropogenic impacts. So this is where we have to start from the beginning yet again. Fernando has been doing a lot of work, as you saw with his ecological surveys, to identify what kind of macrofauna are there. And so now we have to begin to build that reference database, which, as you've already heard, has been really difficult for us up until now, okay? And so just to give you, just to give you a little bit of information regarding the diversity that is found in the Yucatan and Cozumel Island. So there's about 207 species, of which nearly 70 are considered to be stigobionts, many of which are micro-endemics found in single caves, for example. And so even though there is a 400-meter channel between Cozumel Island and the Yucatan, there are 5 species that have been recorded both in Cozumel and the Yucatan Peninsula. So even most recently, we're still seeing more and more records of new species from other phyla. So that was also an emphasis of Fernando's work, to now try to include other diversity besides the well-known crustaceans that are associated with these systems. However, many of these studies still lack molecular data. So during his dissertation, he collected samples from 15 caves, including 8 cenotes in the Yucatan Peninsula, along the Quintana coast, as well as in marine caves in Belize, as well as 5 cenotes in Cozumel Island. And so where we could, samples were also taken from type localities, and also to increase our representation of non-crustaceans. So in order for us to really understand what we're working with, we first need to see what's available in our currently public databases. So he did an extensive literature review and reviewed the public databases to see what barcoding data is available. And then eventually extracted DNA from over 190 specimens, of which 70 achieved positive barcodes. So overall, what does the data look like? What are we working with? So if we go into these databases, we can see that there's quite a number of sequences, and there's quite a bit of bias in terms of the sampling of those sequences, where a lot of it emphasizes crustacean sequences. And so you can also see that there's a lot of sequences. However, 62% of that are focused on just these 6 species that are listed here. So there's still a lot of work to be done. And of the 12 studies that focused on crustaceans, 8 of them either were on remipedes or Tifladia. So as you can see, there's quite a bit of missing information, especially when we're trying to do broader diversity studies like Shari presented earlier. And so from the data that was collected, so he's increased the number of barcoded species, and then representing about 5 phyla, okay? So just to give you a little bit of an example of what are some of the patterns, right? So he generated a lot of data, but I'm going to tell you what can we see from some of the data he's already collected. So this is Anthromesis cenotensis. It is the opossum shrimp. It is broadly distributed within the meteoric lens throughout the aquifer of the Yucatan. And so you can see that it's reported in over 136 cenotes, and the type locality is Ballou. And so when we start looking at the data, okay, so in terms of what was publicly available, the sequence was from Holbox, which is in the Yucatan. And then Fernando added sequences from these 3 sites here, Actunha, Zacatún, and Chacmol. And what we begin to find is that we appear to have at least 3 separate lineages. Now, I'm not here to say that they are distinct species at this point, because again, we're only looking at a single gene fragment, but it's already beginning to show some pattern. So what is important to note that the type locality is located in the center here. So that brings the question as to what is the identity of this Anthromesis cenotensis collected from all the way on the other side of the Yucatan state. So even when we look at species delimitation methods, it does support that there are distinct lineages. So therefore, this is just highlighting that further work is needed for us to better understand what is happening with this particular species and or populations. So we all love remipedes, and so remipedes are also a curious case in this particular situation. So there are two species known from Mexico, Tulumensis and Fucoxburni from the genus Hibagonus. And then we have Kokii from Belize and Cozumelensis from Cozumel Island. Now, Tulumensis, which are indicated, so the type localities are indicated by the stars here. The type locality of Tulumensis is Actunha. So the sequences from the literature were collected from Cape Crustacea. The sequence that was available for Fucoxburni was also from the type locality, but only known from Cape Crustacea. And then we also have the species from Cozumel, from La Quebrada, so the sequence was available from there. And then we also, we also include a specimen, Kokii, from the marine caves in Belize. Okay? And so what happens when we start looking at the data? And so what we find are some interesting patterns. So many of these records, we're only dealing with 4 species here, but these records these species were associated with single caves. However, what the data are showing that they have a much broader distribution. So for example, if we look at Fucoxburni, remember that the type locality was Crustacea. Sampling in Actunha revealed that species occurs there. If we look at Tulumensis, we can see that not only do we have sequences from the type locality, it is found in Crustacea, but the specimen that was identified or described as Kokii is, in fact, Tulumensis. So now, this potentially requires a revision. So again, this still requires more data because we're only looking at a single gene, but already these are revealing some interesting patterns, even with just such little data. So this is just to show you a tree view of the relationships of the sequences we discussed. As you can see that the type localities for Belize and for Tulumensis form a group here, and then we have Cozumelensis and Fucoxburni. And what's also interesting about Cozumelensis, it was only thought to occur in La Quebrada, but it has since been collected from other caves in Cozumel Island. So in summary, this type of work is just a starting point. We are just basically in the beginning stages of generating enough data and being able to identify the diversity that truly exists. But this is really going to be the launching pad for us to establish these reference barcoding databases, but also with the clarity of the species identities. And so therefore, with this, we will be able to use these types of databases for next generation sequencing approaches. Right? So we have the ability to generate lots and lots of sequence data, but if we can't identify most of it, then we're kind of still at square one. Okay, so it's possible that we might need to look for alternate barcoding genes, 16S, 18S as an example. But again, this could be a great way to quickly identify species that are in need of evaluation, either taxonomically or conservation status. So I'd like to thank the members of my lab and all my collaborators and funding sources, and mahalo, and thank you very much.
Maybe time for 1 or 2 questions.
Sure. Kokei is this one. This one. Yeah, so this was the pink one, so Kokei is that.
Therefore, how many sequences did you get?
Fernando, how many sequences did you get?
So I know there's like 9 sequences between the ones of the literature and the news.
Any other questions? Yes.
Can you go back to the last slide, please?
Sure.
The very last one. No, yeah, that one, thank you.